Okay, thanks everybody for coming. I'll sort of try to give a broader overview. Although I have 2-3 slides, but there's some technical aspects for projects that I asked some of the collaborators here. So the first thing I have to acknowledge, everybody here, so that allowed it to make it possible. That let's start with the funding, and I hope I put all the names that are that provided this, which is DPSSC, London Mathematical Society, Newton Gateway, Liverpool Trust, ESC, University of Cambridge, and so on. And then, of course, there are some fellowships. So then the steering committee that decided to accept us to do the program, and Ulrike, which is probably not being able to come, but of course also Mila, thanks for coming as well, and then of course the amazing team which made something amazing, making everything smooth and working. So now, one of the things I wanted to start with is say, okay, what is the program? What is trying to achieve eventually? So what are the complex challenges that we can think about? And I would say that in all the terms we can talk about quantifying response to disturbances is an important, very important topic. Sort of as an umbrella topic that we can talk about. And animal ecology is obviously think about landscape and climate change is the one aspect that comes to our mind. And cell biology, obviously, we have heard values of dysfunctional disease, which is obviously comes immediately to our mind. So just to give examples, we know on the top animal ecology, the loss of biodiversity, we hear a lot about. And then altering signal pathways that promote metastasis, it's about disease and malfunction. And if one, these are very different scales of biology and animal ecology, and then in between that links the two. So I don't have to talk about the pandemics that we suffered through a couple of years ago. So that's actually it's both. So these are just examples that we can talk about. So now, in what else to do movement with all these complex challenges? Well, the first thing that you might think about is that's a very natural way to look at movement as a way to quantify behavior time. And this has to be thought in terms of the recent decades where there have been this traffic revolution. Really, nowadays, we have such a high resolution in terms of spatial temporal data. Then over a very long period of times, which was not possible in the past, and then across multiple individuals. This is key, as you've seen many of these movies about collective behavior. So that's very key. So that's why these complex challenges fit very well to understand how things move. So now, what are the challenges in analyzing the data? Although it's very natural that the dimensions in movement data is tremendous. So we have heard some of the talks that they mentioned this, and you can see the geometry and the stochasticity of the movement is the first answer that comes to my mind. But then it's not just a point particle. You can have lots of points together, which makes a shape, that we heard in the previous talks, that moves over time, so that you have to track that. It's a very different way of analysis, as we heard in the last 2 talks. Then, are we talking about the individual? Are we talking about collectives? Because the interaction becomes important as you analyze that. And then, what kind of interaction? If you know the interaction, or at least you think to know the interaction, what's the strength and the type? So, and you can think of, the spatial range at which 2 objects, 2 entities, living entity interact, and then is it a direct interaction or it's mediated maybe to the environment? There are all sorts of questions that, as you analyze data, you have to try to address. And then you can also talk about the organizational level. So it could be, group, population, communities, and the community can be very different. We heard about phenotypes and phenotypes in cells. No? So it's lots of complexity, and so how do we actually tackle this? Of course, we focus in our activity on some main events, and these main events, I just mentioned them there. So it was the summer school who put us on the same par with everybody, so everybody understood what is mathematics of movement. Then we had, the Rocky lecture from Ian Gooding that excite lots of people, especially in collective behavior. And then we did have, close by a collective behavior workshop, and we have heard all talking about that. So we had another workshop, which is indeed I mentioned before, interaction is very key to understand behavior. It's about indeed how you measure, how you represent interaction. And the next workshop was the one on no Markov. So when the statistics is actually memory dependent, so it's not just the simplest way of thinking about. Then we had also, oh, sorry, I just forgot there is a connection about the pandemic, sorry. So that's a micro to macro in the epidemiological model. Then we had the no Markov. And then there was a quick lecture as well from Becker and on data-driven modeling, which, as you know, it's you can put all sorts of big models, but then how is actually explain the data is very challenging. And I would actually not forget that we had an ECR panel where everybody, not just the early career researchers, but also the academics, were very enthusiastic about, and the EDI panel section of human science. So these are the main events, and in between there were lots of talks, lots of discussion that happened. So we just managed to send out a survey to those that responded, recently, and because of the different kind of areas we covered, so we wanted to know where are they from, no? So what their specialties, and so this is what we got. So now it's not that they had boxes to put in the answer, so we just say, really, tell me what's your area. So now, they put physics and applied maths, which is the most numerous, but I'm sure there are lots of overlapping there. So, and then, I mean, there is the animal ecology. It's quite some big community that came. So they define themselves as experimental ecologists. And then we have also those that actually mention both this discipline like animal ecology. So this is what the answer. And then what was interesting, because of the different type of disciplines, what actually happened in terms of interaction and collaboration. So of those 50 respondents, so they said that they got 62 new collaborators. What it means? So the number one means that there are around 15 of those which were new collaboration, each individual, one of those 15 individuals, and then a smaller number, 4, 5, 3, and 2. So this was for within their own field. But what about across field? Of course, a bit smaller, but it was still impressive to have 44 new collaborating there. So this is just, following on what Paul was saying about the value of this program, is what you actually can achieve, talking about 4 different disciplines, broadly speaking. So now, where did people come from? And so we're very happy to see at the end that they come all over the world, except Southeast Asia. We invited many from them, and somehow it happened. We didn't. I don't know what happened there. But of course, it's also more distant, but So next time we do a better job for that. And now the advantage also coming here at INI, and I should stress that those foreigners that come here, then they were invited to go and interact with people across the UK in other institutions. And so these are all the institutions that we gathered where participants went around the UK to give seminars. So it was quite impressive to see all of these interactions that were happening. So now, okay, let's come a bit better to what we wanted to achieve. So this is like the platform which we could use to achieve what we wanted. And so what did the organizer do besides organize? Also, this probably might be curious. And so this is what we did 2 times a week. We had that little whiteboard there, and we start asking questions. And so this is one of those meetings, if you want. So now, of course, and you can see that being Photoshop. Paul is the one that, because of his, necessity to be at the lab there, so come only 2 times. But so really, we had lots of questions there. We're trying to answer. So that's what we did. So what do we try to go about as we talk to people from 4 different disciplines again? Physics, animal ecology, cell biology, and applied math. 4 different disciplines. Well, there is a big problem in the language. What is common? We try to understand. So that was a really big first step. And that board was really helping us a lot. And so the second steps, once we understand each other's language, is what are the common questions, so the common phenomena. And we managed after some time to, I guess, give you an overview. So we could understand each other by talking about describing what we are trying to study, and seeing that in a ecological setting or in cell biology, you have similar things. Now, yeah. So for example, you can think about in ecology, when you talk about lifetime migration, so that's clearly a synchronized 50 times seasonal movement. and then what's the corresponding one? In cell biology is like, you have a good cell that you revert migration of your flux stream that keeps going through. So, or you can think about structure building, well, you nest and burrows, you think about ants and build nest and so on. And allogenous, embryogenous, really build structure there in the body. So then you can think about collective coordinated, you've heard about it. So block inverting and wound healing for the first talk, for example, about that. So there are lots of parallels there. And then, so we could actually identify them by just going to describing them properly. So now, what was the third step is actually, and that's why we're here, what are the common quantitative tools? So that was probably a bit more challenging, but so now before I tell you what was there, so you have to understand that we actually, based on the visitor that came, we tried to create the set of mathematical areas that underpin all of this. And then it's quite impressive that, okay, we probably cover most of math, I guess, but then you can see from the standard reactive diffusion we heard, to some, stochastic Monte Carlo methods, Ryan walks, topological data analysis, then you go all the way to people like external network, no market, and so on. So this was quite a big area that overlapped all of this animal ecology and cell biology. So now, how do we go about sort of in this cloud? What are the main category of models? So what do we do? So as quantitative scientists, what do we go about trying to create in our mind that we want to model something? So you go through a modeling decision. So, and what that is that you have to decide pretty much, so the individual and the population and how they interact. So you sort of have to think about that. You decide the individual, how does it move randomly? Is being biased towards some direction, or does have some memory of what it's been doing, or and then once you decide what to do, then you have to decide the sort of representation of that movement. You have to use the network, continue the lattice. Then this is the part where you have to decide the level of description. That's also this part are always guided by the data. So are you talking about internal states, like you have disease, you have it or you don't have it? Are you talking about a single individual? Are you talking about a group of individuals or the entire population? And maybe you can actually have models where you worry about all of that, no? So, and the interaction, then it's actually, the environment could be the individuals, other individuals, or it could be the actual environment. So, animal, feeding, plants, and so on. So, now, if you actually try to go and look at papers, you can actually identify, went in and looked at lots of papers, and we identify each of these. We can talk about epidemic spread. You think about, people go back and forth in work and home. Then they see this is actually the classical case where it's modeled as a network. And then here you have the two halves because it's the population, but it's also at the individual levels. And then, sorry, at the internal state level. So, because you're worried about if you have or not the disease. And then this will tell you that you interact with the other or the other within the population. So you can actually go and look at the papers and classify this. This is, for the example, from animal ecology, this is one cell biology. And just to tell you that we really read the paper, this is the example that we picked from that. And so we can actually really extract information. But so how do you actually do the extraction information? That you have to have in mind some sort of paradigm model that you can think of. And what are they? So this is the one which is the more technical. So allow me to make it represent. So there are three formal paradigm. You think about modeling this process in the movement. So you have a Langevin equation, which where we have seen example earlier in the first talk. So this represents the positional velocity of the particle, and it's an index article that could be many. And this, if you want, is Newton's equation, where you add noise in the simplest form. You add an additive noise, and then it's about visible any particle has its own different kind of noise. So this is sort of way to represent lots of uncertainty in this term, in the noise term. So this is one way of thinking, and so you essentially create individual trajectories, and then if you want to extract information about ensemble, like you have to do some ensemble average. Or there is another way, which is actually thinking, instead of thinking about following this individual trajectory, you think about the configuration of one particle having certain properties at each moment in time, which has velocity and position, each of the n particle, and then gives you a configuration. And then you write those objects called master equation, where you look at the probability of that specific configuration. There are an infinite number of that specific configuration that evolve in time through some transition rate with this object, which in the more complex case could be time dependent, complicated enough. So in fact, I mean, if you want intuitively the difference between these two, I mean, they describe the same thing, but what's the difference between these two? You have to think about. One is like, I mean, it's like the infantry on the ground, so you just worry about what you have, and the rest is a novel. That's why you cut with that. And the other one is a sort of area view. You think about all the trajectory together, and you have all the transition rates in mind. And then there is a third one, which is the reaction, what I call the reaction transfer equation, where you talk about mean quantity of this configuration to some extent, no? And so you write an equation, which is now, you don't see here, but that could be, it's a vector, so it's different type of population of the species, it could be the density of the species. And then you have again a transport process, be more complicated than diffusion. This is the diffusive case, and then you have a nonlinearity, and you've seen an example of that. And now again, this is one example of making something non-Markov, time non-local, as we use the term we have seen in the previous one. So again, if you go with that clear in mind, you can read papers and identify where are they, so how to associate each of these models according to them. So, and so this is just to tell you that this is the time non-local. Okay, so now if we come back to the modeling decision tree, so again, so how do we actually go a little bit more in depth here? So we want to expand a bit what we see in the literature. Which one do they use? The bias, the random, the persistent, and then we can also want to understand a bit more maybe the level of description. These are the important part if you want. And so we read hundreds of papers. So this is like what we looked at the ecological process on the left and the cellular process on the right, and the sort of type of model approach that we use. Notice here we worry about the individual, and here the level of description if you want, and the type of also we call it mechanism because it's also the type of process we're looking at. And so what came out, although it's a subset of the huge number of papers you can find out there, that clearly you say all the taxes here, the various taxes phenomena in cell biology, they're all guided and modeled by bias. Bias means probably they have to go in a specific direction. While you see the random movement, only a few cases we spot them, although there is model to random movement. On the other hand, on the ecological level, there are lots of random movement, more than the other one. An interesting other difference, and we noted that environmental modeling, so it's something that is very common. You see here in as we heard this morning about fluotaxis, angiogenesis and so on. You remodel the environment as you move about, and that doesn't often happen in ecology, except in nest building. That we know that's the one case that happened. So there is all of, and this is to sort of see that maybe what we those people working in animal ecology should actually learn from cell biology about environmental modeling, and vice versa. You can see kind of local model. There are a lot in ecological processes, but we only found one in the context of cell biology. So there is a sort of crosstalk that happen based indeed on different aspects. So this is what we actually what emerged. So now we wanted to sort of understand how do you put that together? How can you think in a conceptual way? Yes, we understand the different, but the different modeling framework, but how can you put that together? And so for that, what helped us is the movement ecology framework. So this is something that Ranatan and some of our other collaborators as well. So they put together in 2008. Right? Yeah, 2008. So where actually they tried to sort of conceptualize how do you represent an animal interacting with the environment.
Any organism.
Sorry.
Plants, microorganisms.
Could be also microorganisms. So individual, not in the tissue, not the cell in the tissue, but the microorganism. So that helped us a lot because you can think about the individuals where sort of when you make decisions where to move, has to decide why, where to go, and how to go about how to the locomotion aspect. And so the point is that once it makes this decision what to do, then eventually it moves, and then that's how it affects the environment. Now the environment could be other individuals, it could be the actual environment. physical environment, like eating a plant. So. And the way the environment affects the individual is full-centered. So we have this. So the idea was, okay, so certainly we can sort of look at papers where they measure these quantities, and so we can actually determine the acquisition rate, to sensing each animal has different senses, organs that can be used, and then you can sort of classify and see where they go as you measure this movement response. So if you think about movement response, the acceleration divided by velocity as they acquire information. And so we thought, okay, so this is the way to sort of rather than going in the details here, but then actually understand how to do the coupling and how does that coupling changes in the cell biology world versus the one in the animal ecology. And so that, so that was for us just to find the paper where actually you can describe it. And it turns out we did find some data. Now it's very hard to actually find people that measure the information acquisition rate and measure the movement rate, but we're still in development, but we could find that. So now what's interesting is that, okay, so this is just a subset, this is actually your work. So this is just your data, by the way. So to some extent you expect to be, so the acquisition rate you might expect to be always below the 45 degrees angle. So then you first acquire, you have some rate of acquisition, and then you sort of move accordingly, okay, there could be some delay before you respond, but then the way you try to respond, you just do the same. But actually, if you think about, we are not expecting necessarily that, because you can actually anticipate what's happening. So probably the higher organisms, we might expect to find some up there. So we haven't finished our search rather than being all around. So okay, so this is the game. To finish.
Quantitative biology community is actually a lot of this information acquisition rate, information transfer channel with information amount of information in signal transduction network.
And do they actually study how it transfers to the movement aspect?
Yes.
Okay. So then I want to know about those papers, and certainly that's great. So now, as I told you, I have a couple of examples for given that framework to be a bit more specific. So, I told you about the three-type modeling paradigm. So I've asked a colleague, Guillermo and Philip, to send me something about those two. To Philip, I've asked about reaction diffusion, and then to Guillermo, I asked about something related to the first Landman kind of model. And I was happy that Guillermo didn't send me any equation. Philip did not send me a question, but he sent me a graph. So now I'm in that situation that I have to try to say the right thing. So please correct me whatever I say. Now, here I know that Ran also is been collaborating with Guillermo. And so this is one aspect of the program here, is that we should not underestimate the importance of the experimental biologists here to actually go in and provide the necessary tools for the theoretical ecology, if I may, like that. So this is about the movement of these nice individual animals, and it's about the right. The tortoise, it's 3 nice. So okay, so the way they're trying to model is indeed through an algebra equation description. So the other one that Philip sent me. So this is a collaboration he had with Kevin Painter. That I think they all came here. I don't know Tommaso. I think also Tommaso.
Yeah, Tommaso.
They all came here at the program. So what is interesting? Well, we heard a bit about phenotypic switching from Duncan this earlier. And it's about phenotypic switching in cancer cell invasion, and the way it has been arranged. It's a reaction transport equation. So it's a coupled set of reaction transport equation. In this case, they are also advectives, so they go into the bias. And what was interesting for them to study is probably the effects of the different signaling cues on this phenotypic switching. And so what apparently they noticed is that if you have, you can actually set in the model a constant switching without worry about how it comes about. And so you have these are the plots of the whole population density, and you can see that they have waves, of this population density moving within the same invasion, And if you actually make it extracellular matrix dependent, so the density, I guess, of the extracellular matrix, then you see a big difference in the behavior of this non-propagating and the different amount of population between the two types of phenomena. No? So that, and apparently that could be important to understand if there are evolutionary advantages to having its original population versus not having it. So I hope I did a good job. So now, and a third example, which is the one which is called the Buster equation example. So this is the one that I started, so that's not finished. So with Hernán Laralde from UNAM in Mexico. So these are sort of this broader question which are sort of travel across, animal cell biology. And so if you want to optimize the search of targets, but doesn't matter which one you pick, in the order, the sequential order. So this is something that we start asking the question, can we actually do it? So you can think about, if I'm foraging, you want to sequentially pick the object, not go back to where you've already eaten something. No? So you sort of want to go around, or the immune cells, they want to go in one location first and then another location. So how do you actually go about doing that? So in other words, clearly, if I show you, so if you start here and you have to pick all these targets, and then how do you actually do it? You go about doing this, or do you go about doing the other one? Okay, still in a random path. No? Now, of course, as you can imagine, what depends on what's in the environment. No? And and you start thinking about, imagine what's the environment is a very complicated environment. Here, in this case, you just have two targets, and then you have one, and there is lots of local bias that push it over here rather than over there, but not over there. So these are all sorts of heterogeneity and so on. And then you can start asking the question, what's the time it takes for me to reach here and not there, and vice versa? And then you can see in such a heterogeneous environment, there is a big difference, in terms of the way you go to. So this is a total first pass probability to reach either of the two. But if you concentrate on looking at I reach one and not the other one, there's a big difference. And so that's in terms of optimizing search. It's not the conditional probability yet. This is it's a step before that, but we have developed the format to do it. I haven't shown. Okay, just to summarize. So what is, since I haven't told you what mathematics do, okay? So we have this idea that it's this tool, okay? So there are these conceptual framework that we want to think about that help us. We have observation that's very in animal ecology and cell biology, obviously. And then we have these mathematical models. So this conceptual framework is the one that has to do with living entities. They have to sense the information, there is an acquisition of information, you have to process that information, and then you have to respond to that. And again, the information could be from others, or it could be from the environment. And then the way that it's all linked together in the mathematical movement is because we have this three-paradigm modeling framework that allows us to tackle the different sort of questions. So what are the future plans? Yeah. So well, we want to keep the math movement community engaged. So and the vast overlap that we've seen is present, which we didn't know so much before about the techniques that we use. And so, in other words, we want to minimize the siloed methodology that everybody is naturally working on. And so one of the ideas we have is to organize thematic issues in big conferences, so mini symposia, so that we can keep continuing interacting. And we organize that we want to be convening every 2 times a year, so that we keep the exchange going. And then fundamentally, we want to actually go after complex challenges that already. And then having seen all these things that are happening, we are in a good direction. So these are some of the ones that are already impressed, and some in revision if you are interested. So now let me just tell you what are the planned outcomes that we have. Special collection we were just talking earlier about collective behavior. So that was very popular. And so it probably in December 2024 there will be the deadline for everybody to submit there. Then me and Philip, we are creating an edited book with the same title of the program. So where we have asked, contributing chapters already. And this is what you've seen. I was telling you about this integration. It's a paper that we are preparing, that we, the co-organizers, we're going to submit hopefully soon. And of course, there is a large number of applications which I don't also know about it, which all the participants that came through and are constructing. So let me thank my collaborator very much. So this would have not been possible without them, obviously. So let me personally thank my group and Toby and Ben. So been here for 6 months. They suffered too. Okay, thank you so much. And Alessandro and the others only a smaller amount of time, let's say. So now a thank to all the program participants because it's been really unparalleled experience here. And then I would say a truly intellectual overall. Thank you so much.
And of course, we should have thanks to Luca for really taking the lead and all the work that you've done in bringing it all together. So thank you. Any questions or comments or anything?
I would say that was a beautiful summary,
Yeah.
a short feel in less than 1 hour. So it's amazing.
Yes.
There is this perception, action, loops that people talk about for a long time. So, but they studied in a similar manner or very different? Or
Well,
for its
it's a sort. I mean, it's behind them. There is this sequential thing that really. This is we are sort of trying to quantify it more. So the first step is to try to understand where in that two axes where those cells are compared to others. But you're right. So we are following in that sort of thing. And I guess
that's where the mathematical tools.
Yeah,
are
because
these new tools to study this.
Well, I mean, if to some extent you may not say they are new tools, but it allows us to. So imagine once we have enough data where this animal cell processes are in the graph, we can sort of cluster and see why in some cases some is above the line, some is below the line. Are there evolutionary reasons why they are there? But then we also know which tools, maybe the tools can help you understand where they go. Also, so how to use them. That's the point.
So often you expect there will be domains in this plot of different tools used for different domains.
I have no idea. You can ask him.
No, the point is that
It's possible.
that's what we're after. So are we going to be successful in identify those? See, there is one difficult challenge is that the data on the acquisition rate and the movement response rate is not. I mean, it's very hard, no? So it's and the hope is that really more and more people maybe because it's not that they do that measurement automatically. We have to sort of accept some special cases, blocking and so on, or get data of the plants and so on. So to see, it's it depends.
Work on the dung beetle, it goes some direction, then she climbs on the ball of dung. It looks around, and then chooses a new direction and goes.
Yeah, I
So
mean,
you can really see the acquisition rate.
that could be that could be.
It's very clear.
If you know the paper exactly in mind, which one is it?
What is it, dance?
No, dung beetle.
Done.
Any other? Okay.
I just wanted to say thank you on behalf of the entire Institute. It's been a pleasure to have the mathematics movement program here. So thank you. And you've given us an idea as well. I think all programs should have one of these
The outcome.
sessions. Yeah. So we'll be asking for that.
Yeah, then it becomes a requirement. That's good.
So thank you. And
you.
look forward to seeing all the